Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study published in the Journal of Veterinary Internal Medicine in 2024, which uncovers the genetic basis of a severe platelet disorder in a cat. This research highlights the crucial role of precision medicine in understanding and potentially treating cardiovascular diseases in our feline companions. Inherited platelet disorders, also known as thrombocytopathies, are rare conditions that affect the ability of platelets to function correctly, leading to bleeding tendencies. While these disorders are uncommon, affecting an estimated 1 in 10,000 to 1 in 1 million people, a significant number of variants have been identified in humans, shedding light on the molecular complexities of blood clotting. Glanzmann's Thrombasthenia is one of the most well-recognized inherited platelet disorders, not just in humans but also in small animals. Globally, this condition affects approximately 1 in every 1 million people and is characterized by impaired platelet aggregation and the inability of blood clots to retract properly. In humans, Glanzmann's Thrombasthenia is known to arise from autosomal recessive, loss-of-function variants in either the integrin subunit alpha IIB gene or the integrin subunit beta 3 gene. These two genes are responsible for the formation of the alpha IIB beta 3 receptor, a crucial component on the surface of platelets that is essential for their ability to stick together and form a clot. Similar genetic causes have been identified in dogs with Glanzmann's Thrombasthenia, with certain breeds like Great Pyrenees and Otterhounds showing a higher prevalence. This particular study focuses on a 21-month-old domestic shorthair cat that presented to the University of California Davis Veterinary Medical Teaching Hospital with a history of recurring respiratory distress. Over a period of 15 months, the cat had experienced intermittent breathing difficulties, coughing, increased respiratory effort, and intolerance to exercise. Prior to being referred, the cat had been treated for these signs with various medications, but 4 days before the referral, the situation worsened with the onset of coughing up blood and rapid breathing. Initial examinations at an emergency clinic revealed a low platelet count and anemia. While standard clotting tests like prothrombin time and activated partial thromboplastin time were within normal limits, the low platelet count, along with bleeding episodes, strongly suggested an underlying platelet disorder. 2 days later, the cat's condition continued to deteriorate, with anorexia, small spots of hemorrhage on the skin, and bruising becoming evident. Upon examination at the Veterinary Medical Teaching Hospital, the cat showed noisy breathing and bruising on its abdomen. Blood tests confirmed a low platelet count, but also revealed the presence of unusually large platelets, a condition known as macrothrombocytopenia. Given the cat's history of spontaneous bleeding and the finding of macrothrombocytopenia alongside a low platelet count, the researchers hypothesized that a congenital platelet disorder with a genetic basis was likely. Their objective was to identify the underlying genetic cause of this severe thrombopathia, or platelet dysfunction, in this particular feline patient. To investigate this hypothesis, the researchers conducted a series of tests, including platelet function assays and genetic investigations. Platelet aggregation, the process by which platelets clump together to form a blood clot, was assessed using whole blood impedance platelet aggregometry. The results showed that the patient's platelets failed to aggregate in response to adenosine diphosphate, a key signaling molecule in platelet activation. This complete lack of aggregation was a significant finding, strongly suggestive of Glanzmann's Thrombasthenia. Further analysis using flow cytometry, a technique to examine cells and their surface proteins, revealed a complete absence of the integrin subunit beta 3, also known as CD61, on the surface of the patient's platelets. Since the alpha IIB beta 3 receptor is composed of both alpha IIB and beta 3 subunits, the lack of beta 3 indicated a severe deficiency in this critical platelet receptor. Additionally, the flow cytometry analysis showed reduced secretion of alpha granules, which contain important clotting factors, from the patient's platelets upon activation. Interestingly, the flow cytometry also confirmed the presence of larger platelets in the affected cat, consistent with the macrothrombocytopenia observed in the blood work. Having established a strong suspicion of Glanzmann's Thrombasthenia based on the functional platelet studies, the researchers turned their attention to identifying the underlying genetic defect. They initially screened the cat for a previously identified variant in the integrin subunit alpha IIB gene, known as ITGA2B, that had been found in another cat with Glanzmann's Thrombasthenia. However, this specific variant was not present in the cat in this study. Instead, through Sanger sequencing of the ITGA2B gene, the researchers discovered a novel genetic variant, a double cytosine insertion within exon 18. This insertion, denoted as C.1986_1987 in CC, leads to a frameshift in the genetic code, ultimately resulting in a premature stop codon in the protein sequence. This type of variant is predicted to cause a loss of function of the ITGA2B protein. To confirm that this novel variant was indeed the cause of the Glanzmann's thrombasthenia in this cat, the researchers performed several validation steps. They screened a cohort of 20 clinically healthy geriatric cats and a larger group of 194 unphenotyped cats from the 99 Live Sequencing Consortium for the presence of this variant, but it was not found in any of them. This absence in a control population strongly suggested that the variant was specific to the affected cat and likely pathogenic. Furthermore, by analyzing the DNA sequence of the affected cat, the researchers inferred that the cat had 2 copies of this variant, one inherited from each parent, supporting an autosomal recessive mode of inheritance, which is typical for Glanzmann's thrombasthenia in mammals. To understand the impact of the genetic variant on the production of the ITGA2B protein, the researchers examined the messenger RNA transcript of the gene using reverse transcriptase polymerase chain reaction, or RT-PCR. This analysis revealed a complete absence of the ITGA2B transcript in the platelets of the affected cat, both upstream and downstream of the site of the insertion. In contrast, the ITGA2B transcript was readily detected in a healthy control cat. The absence of the messenger RNA transcript likely explains the complete lack of the alpha-IB beta-3 protein on the platelet surface observed in the flow cytometry data. The researchers proposed that this absence of the transcript was likely due to a process called nonsense-mediated decay, a cellular mechanism that degrades faulty messenger RNA molecules containing premature stop codons. The identification of this novel double cytosine insertion in the ITGA2B gene as the cause of Glanzmann's thrombasthenia in this cat adds to the growing knowledge of genetic variants responsible for platelet disorders in animals. The fact that this new variant occurred in the same region of the gene as a previously reported variant in another cat suggests that this particular area of the ITGA2B gene in cats might be prone to spontaneous mutations. The researchers noted that this region contains a stretch of 6 consecutive cytosine bases, which could make it susceptible to polymerase slippage during DNA replication, leading to insertions or deletions of cytosines. While this study successfully identified the genetic basis for the Glanzmann's thrombasthenia in the affected cat, the presence of macrothrombocytopenia, the abnormally large platelets, remains an interesting and somewhat unresolved aspect. In humans, the relationship between defects in alpha-IB beta-3 and platelet size and number has been complex, with some cases of Glanzmann's thrombasthenia being associated with macrothrombocytopenia due to other coexisting genetic factors. In this feline patient, since the cat was homozygous for the ITGA2B variant, meaning it had 2 copies of the mutated gene, the macrothrombocytopenia is unlikely to be due to another variant in the other copy of the ITGA2B gene. The researchers suggest that another, as yet unidentified, genetic variant in this cat might be independently contributing to the observed large platelet size. Alternatively, other factors such as increased platelet production in response to increased destruction or premature clearance of platelets could also play a role. Further research, potentially involving whole genome sequencing of this cat, could help to unravel the underlying cause of the macrothrombocytopenia in this case. In conclusion, this study successfully identified a novel autosomal recessive variant in the ITGA2B gene as the cause of Glanzmann's thrombasthenia in a cat with severe bleeding tendencies. This finding represents the second reported pathogenic variant for this disorder in felines and underscores the utility of genetic testing in diagnosing inherited conditions in veterinary medicine. Furthermore, the study established a standardized protocol for isolating platelet RNA from small blood volumes in cats, which will be valuable for future molecular investigations of platelet function and gene expression in this species. While the genetic basis for the Glanzmann's thrombasthenia was clearly established, the co-occurrence of macrothrombocytopenia warrants further investigation to fully understand the complex interplay of genetic and physiological factors affecting platelet production and function in cats. This research not only provides critical insights into a rare but significant disorder in cats, but also highlights the potential for precision medicine to improve our understanding and management of cardiovascular diseases in veterinary patients. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.